Thank you. You didn't throw anything at me, but you threw your voice at me, which helped. Thanks. Make sure I'm recording this. So then you've got a CAG4 repeat, which is 12 nucleotides. That's the second chromosome, second molecule of DNA, second double helix. So the two chromosomes only differ in the length of one repeat, 3 nucleotides. Great. Other questions? Sir?
So for this one, there were 2 separate microsatellites.
That's correct. So the question is asking you, and we could write in the second strand of each of these double helices, I just haven't for clarity, but you could. So these are 2 separate molecules. The question, how did we know those were 2 separate molecules of DNA based on the question, the way the question was worded? It's a syntax issue. In the actual mock exam question, what's the clue that we're talking about 2 molecules of DNA? Was there a clue?
Is it the dash?
Yes, the slash between the 2 microsatellites. So in the way the question is written, it says CAG3 slash CAG3 slash CAG4. That's the genotype. And a genotype is, if there's a slash, that separates 2 different molecules of DNA. So you know, one chromosome's got 3 repeats and one chromosome's got 4. More questions?
For something like the metacentric versus like the submetacentric,
Yes.
with the submetacentric,
So what are the total lengths of these molecules of DNA if we're going to draw the agarose gel? So the top one's 19 nucleotides, 22. Everybody okay with that? All right. So what does that look like on a gel? Let's load one sample. Let's make a couple of wells. We're going to load that sample there and the 22 on the right. Now what? What else do I need to do to make this a perfect answer? Okay, so I need MW, which is molecular weight ladder. That's going to tell us how big the sizes of DNA are. It's going to give us an idea. And then a water control. Although because this isn't a PCR experiment, you don't have to show that there's a water control, but that's okay. We can still discuss it because it's important. So tell me what you're going to draw in the molecular weight well after electrophoresis. Where's the positive charge, by the way, on this thing? Positive, negative. DNA is negatively charged, so it's going to be attracted to the positive charge. Right? Opposites attract. Valentine's Day is coming up next week. So the DNA is going to be pulled down the gel towards the positive electrode, the cathode. So what band sizes, doesn't matter what I said on the key, for those of you that looked at the key. What sort of band sizes do you want to pretend are in the molecular weight ladder? 800. Okay, so we've got 1,000. We've got 800.
I would put down to like 25, 20, 50.
Okay, 25, 20, 15. Okay, fair enough. Right? You decide. All right. Here it comes. So 22 base pairs, there's going to be a band about 22 nucleotides, right? This is all relative to, then, those molecular weight standard bands. So companies, this is how we do it in the lab. Come check out my lab sometime. I'll show you some real agarose gel images if you want. We pay companies who make these molecular weight ladders. Biotech companies out in the world synthesize pieces of DNA of known size. They combine them together into tubes. We pay like $125 for a few microliters of this ladder so that we can add it to our gels so that we know exactly what sizes bands we're looking for. So I can buy a ladder that has really small fragments like this if I know that I'm trying to distinguish small pieces of DNA and their relative sizes. Or I could buy something that's more like the top of this gel. It's big pieces of DNA when I'm looking at how big are my big pieces of DNA. So all you need to know is that 22 is somewhere in between 25 and 20, height-wide, right, on this gel. 22 nucleotides will run slower than 20 nucleotides down here, but it'll be faster than or lower on the gel than 25 nucleotides. Okay. So where do we draw the other band? Right below 20. That would be the 19 nucleotide band. And what do we see in the water well? So you haven't added any DNA, so you see no bands. So you just, it's done.
If you do, then you would have to redo the experiment.
Right, if you saw something that looked like that, then you'd say, wait a second, what's going on? There's not supposed to be any DNA in this gel. So then that would make you wonder about that result, especially. Is that actually the chromosome you're looking at, or is that some chromosome that was floating around in the air that fell into your agarose gel and got analyzed by accident? Which happens sometimes, not literally, but contamination happens. Now this individual's genotype is shown there in the parentheses with the slash in between the two chromosomes. So for extra credit, not really, but for extra practice, what would this look like if we actually took that sample from that heterozygote and electrophoresed it in this new well? So we took that person's DNA and stuck it right in there. What does it look like? Okay, they've got one chromosome that's 22 nucleotides long and one that's 19 long. Ignore the fact that I drew those as darker bands. I'm just having fun coloring on my tablet. So you can distinguish one size chromosome from the other. This is what heterozygotes usually look like. If there is a difference in chromosome size, if you have one individual that has both chromosome sizes, you'll see two bands in one lane, one well. Yeah?
Can we go back to number 2 and, okay, so, like karyotyping, the karyogram is the actual image of the chromosomes. Karyotyping is where I'd like some clarification.
Karyogram. That's micrographs or actual photographs of chromosomes. And then what was the other question? Karyotype?
If you want to throw ideogram in there, why not?
And then what about ideogram? Which can be spelled in one of two ways, sometimes with an I, sometimes you'll see it spelled with an E. Same thing. I could do an ideogram right now. It's a cartoon representation of a chromosome. So oftentimes you'll see short arm, if it's a, let's say this is a submetacentric or acrocentric chromosome, you be the judge. You'll see it drawn here with the centromere as sort of a, like you put a corset on the chromosome and you cinched it tight, or a belt or something like that. It's a little bit of an indent in the chromosome. That's how centromeres are indicated on ideograms. So what's the chromosome arm on the top? Is it P or Q? P stands for, depending on who you believe, petite or short. And the Q arm, as I told you one explanation for why it's called Q, the other one is it's the letter that comes after P. It's the long arm. So there's an ideogram of a chromosome. It's a cartoon representation of a chromosome. And I could add on, what am I doing now? So we've got chromosome banding patterns. Those are often represented on ideograms. It's not necessarily just the size and the centricity of the chromosome, where the centromere is on the chromosome, but other information could be added to an ideogram. could have bands, might not have bands. So how is that related to a karyogram?
Karyogram is the actual image.
So karyogram is the picture of the chromosome. So normally you'd get a karyogram first, you'd take pictures of the chromosomes, and then from those you would draw the cartoons. So karyograms and ideograms are related, partly because it's got that gram bit on there. What's a karyotype? Let me see. Let me finish drawing a little ideogram down here. So there's submetacentric or acrocentric. What would it look like if I drew a telocentric chromosome, an ideogram of a telocentric chromosome? Where would I put the centromere? At the top, usually. So it would not have one of those primary constrictions, this part, where the chromosome gets a little bit narrower. So if the centromere is at the top, then these just look like little hot dogs or something. Will their banding patterns be the same or different from the pair I already drew? Could they be the same?
It depends on what state you're using.
So the banding patterns of chromosomes usually distinguish different chromosomes. So this might be, we're talking about a diploid organism. Let's see, that's the 2 copies of chromosome 1. We're talking about metaphase, so are those identical chromosomes or very, very similar genetically chromosomes? Where did those 2 chromosomes come from? Pardon? Not at metaphase. Those are both copies of one or the other. This is after metaphase, happens after DNA synthesis. So you have copying of every chromosome. So when you look at a metaphase chromosome, one that you see with your eye and you say that looks like a chromosome, what I think of a chromosome, those are 2 copies of, say, one, that P stands for paternal now, not arm length. So an ideogram could also show the same chromosomes, same centricity, and usually the same banding pattern, but now where did these come from? This is how we know these are homologous chromosomes, circled in blue, homologs. They're all chromosome 1s because they all have the same banding pattern. The banding pattern is each chromosome's unique fingerprint, if you will. So where did those chromosome 1s come from? They're not paternal, they're maternal. So that's the copy of chromosome 1 you got from your mom. So the banding pattern tells us which chromosomes are related to each other, which ones are both chromosome 1 or chromosome 2 or chromosome 7. Over here, I'm starting to draw a second chromosome, so let's call it chromosome 2. So if you looked in a metaphase cell, there would be 4 double helices, and they'd have a different banding pattern. And the banding pattern for all of those would be the same. And that's how we would know to cut and paste those pictures of those 4 different molecules of DNA together to call them chromosomes 2. 2 from mom, 2 from dad. So now we have an organism that's got 2 different chromosomes, chromosome 1, chromosome 2. What's the karyotype of this? Two n equals 4. If you do the division, what's n? Algebra. Do it. N equals 2. And n, bless you. What does n mean? How do we know what n is? The number of pairs at metaphase is the number of distinct chromosomes. It's the number of different chromosomes. Right? So here's one chromosome. It's got a banding pattern where there are 2 bands on the short arm and 4 on the long arm. I defined that up here as those are all chromosome what? Chromosome number 1. Okay. Here's chromosome 2. This organism has only 2 chromosomes. There's chromosome 1, there's chromosome 2. Regardless of how many different numbers of copies of each of those chromosomes, they have 2 different chromosomes, 1 and 2. That's what N is. And that's the karyotype. 2N equals 4. So that 2 in front of the N means that it's a diploid organism. When you look during interphase, that cell has 2 copies of every chromosome. So it's got 4 total chromosomes during interphase, 2 copies of each of the 2 chromosomes. So the grams are images, and the type is a written description of the karyogram, or the ideogram.
But it doesn't give you as much information as the grams.
It doesn't. It just tells you how many chromosomes there are. Yep. That's the karyotype. Karyotypes can get more complicated and give you more information. We'll get into that later in class. Yeah?
So we can't just write n equals 2, we have to write 2n equals 4.
The only reason to do that is what's the difference between saying n equals 2 and 2n equals 4?
It could be other numbers with the same fraction.
So numerically or mathematically it's the same, but the 2n is important. So what's
Yeah, it tells you if it's diploid or...
right? So what's n for humans? We're 23, so our n is 23, but we refer to our species as 2n equals 46. So the 2 is important because it tells us we're diploid. So if we were studying salmon, for example, you would want to know that 4n equals whatever. From that, you could calculate how many different chromosomes are we going to be talking about, but the 4 is important because it tells us ploidy. So if we just said every organism is n equals 1, n equals 2, n equals 3, that would be fine, but we'd also have to add some information about how many copies of every chromosome does each cell have. So we just use this 2n, 3n, 4n, and so forth nomenclature.
I found number 3 out way more complicated. I mean, I got it none, but I complicated it like crazy.
Number 3, complicated like crazy. Let's see.
I turned it into like crazy math.
Yeah, number 3 doesn't have to be a crazy math problem. But Chargaff's rule. A's and T's and C's and G's. Should we do another one just for fun? Or for practice, rather, regardless of whether or not it's not fun. Let's see. I'm not going to make one up exactly like that question. But there is a strict mathematical relationship. A's equal T's, C's equal G's. So let me think about this for a second. Think of other questions while I'm thinking about a good question to ask to help you practice Chargaff's rule. Whoa. So other questions while I'm playing around with this?
Yeah, number 9, this BAM HI, it cuts at GATC, and I found GATC twice, so I actually cut it twice.
We can definitely talk about that.
So I wound up with 3 fragments, with all of them with like sticky ends.
So, timeout. BAM H1. What's the recognition sequence? how long is the recognition sequence of BAMH1? 6. GGATCC. So let's back to another Chargaff's rule practice. This is very similar to the one that was on the practice exam, just slightly different. So say down here at the bottom you've got a fragment of DNA you know is 60% A's and T's. So which of the following molecules could be the complementary strand to the strand that you sequenced? So those of you that are counting, what are you doing right now? Help everybody else out. How do we solve this problem? Yeah, you count the number of A's and T's, so there's 6 out of, how long are these? I made them all 10 nucleotides long, so that's 6 out of 10 or 60% A's and T's. Now, of course, on the exam, good test-taking strategy. I know you're all upper division students now, so you probably already know this, but what do you need to do after you see that A seems to meet the requirement for the answer? You still check the others because who knows, maybe there are more than one right answer. Okay, so here's 80% A and T, which can't be right, and another 6 out of 10. So either of those two could be complementary strands. If one strand has 60% A's and T's, then the other strand will also be 60% A's and T's. More advice, of course, when you're done, when you turn it in, double check all your work. I hate to have to give, which you could have an easy response to, well then why do it at all? I hate to give less credit because somebody skipped over part of a problem. I tend to ask multiple part questions. So read the question carefully, go back and make sure you've answered every facet topic in the question.
If we have a question like that on the test, would we be marking, mark both of the answers?
Not necessarily. Again, because my philosophy is, do you show understanding of the concept? So by choosing A and not C, you would still show me that you know what you're doing. You would just show me that you weren't totally effective in executing the complete answer. So maybe a half point or a 3/4 of a point off or something like that, if that was a 2 or 3 point question. But in general, again, I'm looking for your understanding of the material. I'm not trying to be an asshole. So regardless of what you might think later, this is not a game. It's just me assessing your understanding of the work. And that's how I try to write questions, is to get differentiation between who knows a lot, who knows a little bit less, who knows a little bit less, so that we have a spectrum. Other questions?
So you said it's up to us if we want to bring a laptop or a tablet, right? Or does it all have to be on the tablet?
Tablet would be better.
Because I mean, there's this one. I said metacentric, or this number 2.
Yeah.
And I mean, like the P's and Q's kind of look similar. So,
All right. So how do you tell a metacentric? What's the definition of a metacentric chromosome? Its centromeres in the middle, meta. Okay, so if I drew a metacentric chromosome, what would it look like? A pair of metacentric chromosomes would look like an X. The centromere is where the two chromosomes, touch. Submetacentric is slightly off-center. Ooh, I'm a scientist and I said the word slightly, it's sort of qualitative. And that's the root of the question, that's the problem, is how do you as a student know what I mean when I say, tell me if this is submetacentric or acrocentric, which looks maybe like that. What is a telocentric chromosome pair, two chromosomes that are telocentric, what are they going to look like at metaphase, which is when all these are being drawn?
It basically has no centromere, like it's at the very.
Well, it has a centromere, but it's at the
It's at the very end.
very end. So it would look like an upside down V or a right side up V, depending on. So that's telocentric on the right. Metacentric on the left. Those are easy, right? Centromeres at the middle, centromeres in the end. And then we've got a continuum in between those two. Here's where the good news comes. I'm flexible. If you call an acrocentric chromosome a submetacentric or a submetacentric and acrocentric, I don't care. Because we haven't talked about the precise mathematical definitions of exactly how far off center the centromere has to be. So to me, as long as you demonstrate understanding of the principle or of the definition of what a metacentric, submetacentric, acrocentric, telocentric is, that's what I'm looking for. Do you understand the concept? So if I asked you to identify, sorry, that's ambiguous. If I asked you to identify chromosomes by centricity, I would be happy if you called something that looked like either of those two in the middle, submetacentric or acrocentric. You could apply either label. It's fine. Yeah. Number 7. How many number 7s were there on this thing? Draw the agarose gel results you would observe. Okay. Since you asked, here is what's going to be at the top of your exam on Friday. So these are the details, which you probably can't read, so I'll read a little bit of it for you. This is on the lecture slides as well. I posted this PDF with this slide already, so you can look it up later. But one electronic device, these are the highlights. One electronic device, you can't have a smartphone and a tablet and a laptop all going at the same time. You can bring paper and pencil if you want to do some offline doodling, scratch paper, blank paper. If you want to do some work on the side and then just write the final version on the tablet, although the nice thing about the tablet is you can erase pretty nicely without having to have scratch paper, also better for the environment. But it's up to you. So you may use blank paper. You can access any of your own digital notes, the course textbook, and other resources on the internet. It's open internet exam. Because I can't ask you to do stuff on the tablet and expect you're not going to be tempted to go Google something. So I've written Google-proof questions, basically. You're not going to find any of the questions I asked. You're not going to be able to copy and paste the question into Google and get the answer. However, you are not allowed to either vocally or digitally collaborate with other people. And it's hard for us to detect, but I ask you not to do it just because in the context of an exam, that might be considered cheating, since I've specified that I ask you not to do that. Normally I like group work, but on an exam, sometimes not. So if we catch you doing that, that's one of those things that's down at the bottom that says failure to follow the above instructions could lead me to invalidate your test, which means you get a 0. So if you don't follow all of these stipulations, you're putting yourself in peril. So one electronic device, you can't use audio because that could be distracting to other students. So you could go back and watch a lecture video if you wanted to during the exam, if you thought that would help. You just can't use the audio. Because even if you have headphones, it doesn't matter. No, you can watch, but you can't listen. And then down there at the bottom, again, I've said you should screenshot as you work. Take screenshots of your tablet screen just in case something bad happens. And then below that, the most important thing I can tell you, and then we'll move on with other content questions if you want, is check your mail.fresnostate.edu account regularly all afternoon after the exam, because what we'll do, Fatima and I, will go through and look to make sure we can see your edits after you submit it. And if we have an issue doing your exam, we'll ask you to then attach some screenshots or resubmit the exam. And if we do email you and ask you to do that, we expect a prompt response. So you don't have to sit there hitting refresh, 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 refresh. It's okay, but we would like to have the exams, if there's a technical issue on our end, by the end of the afternoon. Hopefully that's not stressful, hopefully that's actually stress reducing, but I don't know. Go ahead.
You said we could bring blank paper, but can we bring like paper that has our notes on it?
No.
Paper?
Blank paper only. Yeah, blank paper only, no written notes. Digital notes fine, written notes no. The point here is to emphasize that not only is this a tablet class and you might be expected to be taking notes on the tablets, I don't care how you take notes, you can take notes on paper or not, but the point here is to reinforce that there's some things that you just need to know and have in your memory, and you can choose to stop editing the PDF and then switch to a different app to look up the notes or to watch a video, but that's going to cost you, of course, time that you should be spending answering the questions. Right. So it's a little bit of a carrot to try to make you remember or understand that it's important to actually study and have some of this stuff stored in active memory for use. Blank paper only. Other questions? Yeah.
I have a question on the privacy.
Yeah, please.
Number 5, for C, but substitution, would that work?
So, B5. Let's see. Question 5. You said B?
C.
C. So TGAC, TGAT. So there's a T to C, sorry, an A to C SNP, single nucleotide polymorphism. You're asking if it's okay to say substitution. I know what a substitution is, and that works for me. It's also a single nucleotide polymorphism. Substitution, though, I'll just caution you, has a different meaning when you get a little bit farther along in genetics and especially in evolution. So just so you know, you heard it from me once. Substitution does work in this instance, a substitution of one nucleotide for another, mutation. But there are additional fields of biology where that is in a different context.
Number 4, there's more than 2 differences, right?
How many differences are there between DNA and RNA? What are the main ones? So there are more than 2. Okay, so ribose versus deoxyribose. So let's see, DNA, RNA. Well, let's see, there's the fact that there's a D and an R, but you have to actually, thank you. You have to actually explain, you can't just say that. There's a difference in the way they're spelled. But if you tell me what it means that they're spelled differently, like there's a different sugar, there's one more oxygen or one less oxygen, that's what I'm looking for. Okay. Which one's single-stranded, which one's double-stranded?
Well, in eukaryotic organisms,
Single-stranded, double, except for, right, there are exceptions. So again, you have to be careful, hold on. You have to be careful when you answer these to show me that you're explaining the answers, not just giving me answers. Is there such thing as single-stranded DNA?
yeah.
How would you make single-stranded DNA? Heat it up. You make single-stranded DNA. So I'm a little bit unclear whether or not single-stranded versus double-stranded is by itself would be a good answer to that question. I mean, it is, but RNA is usually single-stranded, but we also heard, can RNA be double-stranded? RNA is normally a single-stranded molecule, but it can base pair with itself to form hairpins. So be mindful of the exceptions that you know about. What are other differences between DNA and RNA?
Cell versus thymine.
So there's a third one. These are the main 3. Strandedness, deoxyribose versus ribose, and DNA contains thymine, the nucleotide, while RNA contains uracil. Yeah.
What about structural differences? What if we were talking about the processes that they do that?
Sure. We haven't gotten there yet. So I don't expect you to say there's a difference between what RNA and DNA does in the cell, but yes, that's where we're headed in class. Totally valid. DNA is the code that encodes RNA. They're not equivalent molecules, for example. And if anybody's interested in finding out, not now, because you're focused on something else right now, we can talk about why our two nucleic acids have thymine or uracil, but we haven't settled on one. You know, it might just seem to you as sort of a trivial, annoying little thing that one molecule has thymine and one molecule has uracil, but there's actually a real functional difference between why a cell might actually care, if a cell could care, that its DNA has thymine and its RNA has uracil. We'll get there.
Uracil can basically care what, like, belonging to this.
There's all sorts of weird things that uracil does that, yeah, that are important. But we haven't gotten there yet, so that's not on the test.
Number 10, math. More math.
Number 10, more math. Would anybody like to ask any questions about number 10? That was this one about the restriction site that's got a bunch of N's in it, and how frequently we would find that in the genome. Yeah?
Do we have to actually calculate out the number, or do we just put like 1/4 to the 6?
As long as you think that showing your work is important, which is one reason maybe not to do scratch paperwork and then submit only answers on the PDF. If I can see your work, or if Fatima can see your work, that's helpful. If you know, for example, especially if you've got soon enough, thank you for saying that. If you've got a restriction enzyme or any sequence of nucleotides, I just made one up. So what is that? 10 nucleotides. This restriction enzyme only cares about those sequences and that they be 4 nucleotides apart. So it doesn't matter what the ends, what the nucleotides in the middle are. The restriction enzyme doesn't care. So the question is, if you already know that the probability of getting any nucleotide there is 1 out of 1 or 100%, and the same is true for those 4 middle nucleotides, then do you have to show that work? Not necessarily. So in this sort of a case, if you get the right answer, you get the right answer. How many nucleotides are in the recognition sequence of this enzyme? How many specific nucleotides is it actually looking for? 6. It's really 10, but those 4 in the middle don't count. So the question was, if you know that the probability of getting each of those 4 nucleotides is 1 in 4, and if you know you have to multiply all of those together, can you just jump straight to saying you know that it's 1/4 to the 6th power, 1/4 times itself 6 times is the frequency? That works for me. For a simple mathematical question like that, if you get the right answer, you got the right answer. So 2 important points for you to remember. N's don't count when you do these sorts of calculations. And it's not as complicated as it looks when you see 10 nucleotide sequence if there's some N's in there. All right, we've got 5 minutes left. Since no one's asking a question, I'm going to ask you a thought question. What if this was a 7 nucleotide sequence and that 7th nucleotide was either A or T? You've got a 1 in 2 chance of any one nucleotide being A or T. So then the answer would be 1/2 times 1/4 to the 6th, because now there are 7 nucleotides here in blue that are part of the recognition sequence. I wouldn't do that to you on an exam. We haven't covered that in class yet. Yeah, exactly, extra credit.
What? Can you do that again? How come it's, okay, so we have 7 nucleotides that
Yes.
it recognizes, right?
Okay,
But, it's only 1/2 because it can either be A or T?
correct. Yes,
Whereas
so
C, C,
It's
a 1 out of 4 chance.
one of the 4 nucleotides is required there. 2 of the 4 nucleotides, or 1 out of 2 nucleotides, is required that site for that enzyme to bind there. Thank you for asking. It's an important point to be clear on. And there are, I'm not just making this up, well, I am, but I'm not. There are enzymes that are kind of ambiguous about what nucleotide they're looking for. So some of them will have a single nucleotide. It has to be a C or it has to be an A, as we've seen initially in class. Some enzymes don't care, that's the N. And some enzymes do have a preference for a purine or a pyrimidine, but not both. So what's a purine? Adenine and guanine. And pyrimidines are the ones with the Y's in the names. Thymine and cytosine. All right. Now, last thing before you're definitely going to want to hear this. Because if you don't, then you're not going to get all the points on the exam. Worked pretty well. Thought it was going to work faster. Here's a link to a video. Watch the video before the exam. There's a question on this video on the exam. And you're not allowed to use audio during the exam, so you better watch it beforehand. It's short. It's 1.5 minutes or 2. It's not for me either. It's somebody else talking. So that's it. Do all of this. And lastly, remember, if you haven't been doing this already, make sure that Fatima is on your group Google Doc. If you don't have her, you're getting 0 points in the group. Office hours now for 1 hour, and then I'll see you on Friday.